Greetings. My name is Cheetah Chudy, and I am a farmer, rancher, consultant, and applied mycologist based out of Northern California. The purpose of this video today is to give you a primer and an introduction to the types of toxins that are generated in wildfires and urban firestorms, and ways that we might potentially mitigate these using the bioremediation potential of fungi, also called mycoremediation. To give you a little bit of a background, been farming and working with fungi for about 15 years. Back in 2018, my farm was burned to the ground during the campfire, prompting me to launch a program called Butte Remediation. So this program, we basically go out into the community and find underserved individuals, those were left behind in the cleanup program, and test the soil for different contaminants, and then apply fungi to the soil and to the environment to try and retain, mitigate, or otherwise immobilize the toxins generated in these fires, and then retest after the process to see if we've made any sort of progress in remediating these contaminants, these environmental pollutants. So we'll just go ahead and jump right in. So fungi, they're well known for their ability to reduce erosion, build soil structure. They produce these extracellular exudates, these glomulins that can help stabilize and create water-stable aggregates. And beyond that, their ability to degrade wood is functional potentially in the mitigation of some of these toxins. So briefly mentioned, my farm was destroyed in the campfire. The campfire at its time was one of the most deadly and destructive wildfires that we've ever had. Now, in the great year of 2020, we're experiencing even more urban and wildfire storms that have set a new precedent. And so at the time, I was focused on the campfire, but now this research can be perhaps applied at large to some of these other fire-affected communities. The campfire was something otherworldly. It swept through the town of Paradise in just a few hours, fundamentally leveling it to the ground. 19,000 structures and 86 people were killed in the process. And somewhere in that process, my own farm was lost. So this is what I came home to. These are some images of my own farm. And so pretty immediately, just on basic observation, I saw that there was going to be widespread contamination. Here in the lower left-hand corner, you can see this river of aluminum coming out from this burned vehicle. There's a battery under the hood, which presents the potential for lead contamination. Upper right-hand corner, this is my pump house where my water distribution came from. You can see here that those are what used to be water tanks that had melted to the ground, so melted plastic everywhere. In the foreground here, aluminum ladder. So when this massive disaster went down, basically FEMA got involved, Cal OES, the California Office of Emergency Services, for that debris removal. Fundamentally, you were not permitted to remove the debris from this fire by yourself, with the exception that if you had lost a structure smaller than 120 square feet, you were on your own. So if you were living in a trailer before, perhaps you lived in a tiny home, something along these lines, you were on your own for this debris removal. And this specifically was the community that we set out to target with this program, is find people that didn't have the resources to get this debris scraped up, get it removed, and fundamentally were left living with this garbage. So some more images of my home. When the fire swept through, it destroyed all the vehicles, my house, our barn infrastructure, and our mushroom greenhouses you can see here in the lower left-hand corner. And while the greenhouses had melted, the fungi themselves had actually survived. The mycelium within these bags was still alive. And so looking around, I saw this widespread contamination, was somewhat aware of the bioremediation potential of these fungi. So I went about creating furrows, these downhill sloped areas and scraping them back with the tractor and taking the surviving mycelium, where you can see here after a rain event, even though there's melted plastic dripping all over the bag, the fungi itself is still alive. So I built these biological barriers, these wattles, they're called, these straw tubes inoculated with fungi to try and mitigate, try to minimize the spread of this toxic ash in the rain events that came directly after the fire. So upper left-hand corner is my home, and specifically this corner of the home was my laboratory where I grew mushrooms for our commercial production. And so as part of my path to recovery in rebuilding our farm, as well as starting this program, I built this mobile mushroom facility. So it's a cargo trailer and it's got all the equipment required to grow fungi. And seeing the damage in my community and understanding that there was going to be a lot of unaddressed contamination, I set about launching this nonprofit effort called Butte Remediation. So understanding contamination, this is a slide taken from some folks at Chico State that were doing sampling directly after the fire event. And while we somewhat understand the dynamics of general forest fires, urban firestorms are much poorly understood. So this slide, also taken from those same folks, is just talking about what happens in these fire events. We have these heavy clay soils that are then fire affected. They are baked just like a clay pot and become hydrophobic. They will not absorb water. And so we have a lot of surface runoff. Also, we get this DOM, which is dissolved organic matter, and it becomes a carrier for some of the heavy metals and other contaminants that are released in these fire events. We then get a rain event, and this topsoil, because it's hydrophobic, everything runs off the surface, then enters the hydrologic cycle, and now it's in an environment that it's impossible to remove. It's entering biology, returning back into the soil strata, and it's much more difficult now that it's entered the environment to remove or remediate. So again, soil organic matter, it becomes a transporter, what we call a chelator of these contaminants. And so the soil particles serve to be a carrier for the contaminants that we're concerned about. So when it came to the debris removal program, they were focused on these 17 heavy metals specifically. In addition, there's different, what the EPA identifies as persistent organic pollutants, and we're going to talk about that more. There's also asbestos, sewage contamination. There's also widespread contamination of the water system due to the burning. Here we're just a snapshot, again, from these folks at CSU that are doing the work. We can see in this vehicle, there's all these potential sources. We have the lead from the battery, the copper wiring, the wheels have melted off, the radiator melts, and even the tires themselves, when burned, become a persistent organic pollutant. So any given number of contaminants that are potentially generated for a single fire event. So here we're going to break it down. There's basically two different forms of persistent pollutants that we're going to talk about: persistent organic pollutants and heavy metals. And we have different strategies for dealing with these things, and many of them are overlooked. And I think that's something to think about going forward as we see the new future of more widespread fire in California and across the country. So persistent organic pollutants. These are an organization of carbon and hydrogen molecules, and sometimes chloride or bromine, that are organized in such a way that they present a risk to health and human safety. So here, front and center, we can see the benzene molecule. And what this is a ring of carbon molecules with these hydrogen arms coming off of them. Benzene was a big deal in the Paradise fires because the whole water system was contaminated with benzene. When basically the whole city burned down, the water system depressurized, and all the burning plastics and paints The vapors from that were absorbed back into the water system, as well as one of their distribution systems. Their reservoir melted and burned, and so they had these benzene molecules slipping into the water system that way. These are very tough, very recalcitrant, persistent molecules. They'll go on for a long, long time. They can bump into your DNA and contribute to your risk of cancer. And so that's why we're really concerned about them. There's a lot of different manifestations of these benzene style molecules, and we'll talk about them specifically in a second. The other contaminant that we're concerned about are heavy metals. So these are individual atoms. These are ions. They cannot be broken down further. And fundamentally, what we have done historically is we've extracted these ores from the earth. We've refined them for our uses to make our automobiles and our homes and our structures. And then when they're struck by fire, they become free again. They become loose molecules that can go into the environment and create a host of problems for health and human safety and the environment. So we're going to talk about persistent organic pollutants first. These are the benzene molecules, what we're talking about. So benzene itself, I'm not going to talk about this in depth. You can pause the video and take a look at this slide yourself. But again, it's this fundamental subunit of a lot of our contaminants. Carbon and hydrogen atoms that on their own are not terribly dangerous. I mean, they're the building block of life, but these are organized in such a way that they are detrimental to health and human safety. These hydrogen branches coming off of it means that the molecule is hydrophobic, does not absorb to water well. And so bacteria, the type of organisms that would normally break something like this down, are able to attack it. And that's where fungi come in. Dioxin. Dioxin is really bad. It's very much persistent in the environment. EPA says it has a half-life of about 8 years. It is toxic in very, very small quantities. It likes to lodge in our fatty tissues, again, contributes to cancer. There's also furans, F-U-R-A-N-S, and that's basically a dioxin with one less oxygen unit in there. So the branch is just shut at the bottom. Polycyclic aromatic hydrocarbons. Perhaps fungi are most known for the breakdown of PAHs, as we'll call them going forward. Polycyclic aromatic hydrocarbons, most notably in petroleum products. Like when you hear about fungi breaking down motor oil, we're talking about PAHs. And so again, when you burn plastics and other material like that, any of our hydrocarbons that go up, any of our fuel that burns, it's going to make virulent PAHs. Polychlorinated biphenyls, I haven't encountered this a lot in the soil testing that I've been doing. I know other people have found it. There's also a bromated version, so these chloride branching molecules also have like bromines on them. A lot of times from cleaning products when it comes to the polychlorinated bromates or biphenyls. Heavy metals, again, so these are just atoms. They are ions, cannot be broken down further, but a lot of them in levels that are too high are disruptive to our body, our endocrine system, create cancer, what have you. Also substitution. For example, aluminum behaving like calcium settling in your bones and brains. So we're going to talk about just a few of them. So arsenic, I honestly haven't come across it too much. There is some circumstances where we see naturally occurring arsenic in our California soils, and it can be exacerbated by fire. Basically, our green serpentine soils have a naturally occurring form of this arsenic. And so it can be an instance, but again, not a fire-borne toxin from the soil testing that I've been doing. Barium, I've seen more than I thought I would. Not a critical contaminant, mostly noteworthy for aspiration. If you breathe it too much, it can be a big deal. but we can absorb quite a bit of it into our body and be okay. But it is more prevalent in a fire event than I thought initially. Aluminum. Aluminum is widespread and in high quantities. It's dangerous because, yes, it behaves much like calcium in our bodies when we do absorb it, and is, I say loosely linked, but it's pretty well linked to Alzheimer's disease. So a long-term health effect there. Lead, widespread and common. Most noteworthy, batteries. We all got them. Every time a vehicle burns, you see a puddle of lead underneath it generally. Cadmium, another pretty common one, mostly associated with vehicles from what I've seen. And in that same vein, chromium, often associated with burned vehicles from the chrome detailing that's on the vehicle. Mercury, not so prevalent. I thought it was worth adding in, but not a super common fire contaminant, but definitely liberated by fire. Antimony, not a commonly known heavy metal, but pretty common. Mostly used for hardening exteriors of metals for things that need to move, especially like bearings or automotive parts, things that need a lot of metal on metal friction going on. That's where you see antimony. So fungi, where do fungi enter the picture? So fungi in our world, they're powerful decomposers. They break down all the organic matter that from anything from a leaf litter to fallen trees. Fungi are the engines of decomposition in our world. The fungi, they help, as I said before, improve the porosity and the strength of soil, its ability to hold up to damage from raindrops, what have you. And so when healthy soil has fungi growing through it, we get good infiltration of water instead of just making mud puddles. And again, a fire event is going to make these heavy baked clay soils that are not going to want to absorb water. There's a lot of research in the laboratory settings of fungi breaking down persistent organic pollutants and uptaking heavy metals, but very little in the field research has been done. And so that was one of the big goals I had setting out, was to try and take things from the laboratory and do some of the comprehensive soil testing that's necessary to see if these fungi potentially, even maybe, can help, as a lot of the hype suggests. So to take a step back, what are fungi? So fungi include saprophytic. So these are the organisms that break down debris. They only eat dead stuff. Symbiotic. This is a fungus that works in association with another organism, specifically plants or algae. So 80% of the plants in the world have a mycorrhizal association. This means a fungi that grows into the roots of the plants, and it extends the root zone of the plant. It helps to uptake in nutrients, specifically phosphorus, especially phosphorus, and has other auxiliary functions like fighting off competing fungi and kind of making these common mycorrhizal networks that have demonstrated the ability to communicate between plants, as well as some sort of energy flow between plants to help the understory canopy survive in a dense forest. Parasitic fungi. Parasites have their place in the world. These are the fungi that attack living organisms and steal their sugars, steal their energy for their own use. So those are the major three groups of fungi that we have in the world. They're eukaryotic organisms, which means they have a central nucleus. So they're more similar to us than they are to bacteria or plants. They're filamentous. They grow in these thin threads that you can kind of see in the background of this image. They have these single cell networks that aggregate and grow out through the world, but they're not always filamentous. Yeast being the prime example there, which grow in individual cells that are free floating. They reproduce by spores, sometimes not always. So fungi, all three of these lifestyle habits, the saprophytic, symbiotic, parasitic, they're all heterotrophic, means they use somebody else's energy. They use photosynthesized sugars that have been created by somebody else as their food source. And so it's through these 3 different lifestyle pathways: eating dead stuff, helping somebody out, and sharing sugars, or just stealing sugars from somebody else, that fungi make their living. And it's widespread. I mean, it's the most common thing in the world. We're breathing mold spores as we speak now. So this is kind of the generalized life cycle. It's not true to all fungi because they're far too diverse to kind of boil down. But when we see a mushroom in the woods, this is just the above ground portion of the organism. Underneath that mushroom, there's a large mycelial network, which is a term we're going to use a lot, mycelium. So basically what we call this is the fruit body. It's the reproductive organism, and its reproduction is spores. So when a mushroom cap expands, millions and millions of spores are thrown from the underside of that cap, from those gills or from those pores. These spores fly out into the environment, perhaps into the upper strata of the atmosphere, perhaps land right on the ground, and they germinate. And something unique in fungi is you get 2 different possible mating compatibilities. So they grow through the world as haploid individuals. 2 hyphae of compatible types cross. They exchange genetic material. And then from then on, they grow on as a complete sexually capable organism. When they've amassed enough nutrients, or perhaps things, the going gets tough, they decide or start to make a mushroom again as a method of getting out of Dodge or procreating via their success. But then the mushroom itself is formed and the cycle starts again. So I just want to throw this in there to say I'm generalizing fungi a lot for the purposes of this. We're just talking about saprophytic fungi. Again, those fungi that just eat dead stuff. And there's far too many forms of fungi. There's far too many lifestyles. There's far too many habits to go into on this. So we're kind of narrowing our focus here. This is a laser focus on just those saprophytic fungi that are capable of helping us in the scenario. So fungi eat wood. Okay, so how can they help us? Fungi eat wood, and primary components of wood is cellulose and lignin. You know, there's some hemicellulose and other stuff in there as well. But on the left side here, we see cellulose. It's this very simple, linear glucose subunits linked by an oxygen linkage. A lot of fungi can break it down. A lot of other organisms can break down cellulose okay. On the right-hand side here, this is lignin. So lignin provides the structural, tensile strength to wood. And it's this irregular, bony, tough molecule that a lot of organisms don't have the ability to break down. Okay, so here we're looking at, on the left, the benzene molecule and the lignin molecule on the right. So here we can see this chemical similarity. That same benzene molecule can be found in the lignin molecule. And this is where we come to the potential for fungi to help us degrade these persistent organic pollutants. We can use the enzymes typically used in the degradation of lignin to break down these persistent organic pollutants. Heavy metal uptake. So there's research to indicate that heavy metal, in the same way that we can accidentally absorb it and it becomes problematic for us, fungi can absorb it as well, but they have some biological stratagems that are much more suited to the task. They're much more flexible in ways that other organisms aren't. And there is some research to indicate that they will cytoplasmically stream these heavy metals into the above ground portion, the mushroom itself, which is, from the fungus's standpoint, a way that they can basically take the trash to the dump. It can be streamed into the mushroom, which might be taken away by some unsuspecting human or deer, and taken out of the path of its future offspring that are most likely going to spring up in the wake of this mushroom. And I'll add the caveat that I have not seen a lot of that translocation of heavy metals up into the mushroom. I just don't think it's a functional thing for bioremediation, but we'll circle back around to that with some hard data. So again, fungi uniquely well suited to the heavy metal task in ways that some organisms aren't. So upper right hand corner, this is a scanning electron microscope photo of mushroom mycelium. This is work done by a group out of India. And so normal mycelium in the above image, and in the below image, this is mycelium that's been exposed to heavy metals. And what the fungi have done in this study is they create these compartments. They build extra vacuoles, which vacuole is typically used for lipid storage, fat storage. And they create these extra storage compartments for holding on to these metals. So standard bioremediation, you're basically shoving it into the plant and then removing it later. Mycoremediation is a similar thing, but the fungi are a little bit more tooled to the task. So different fungi, based on the research, based on the literature out there, are more or less suited to the task. And so we're just going to take a second here and talk about a few of those species that are readily cultivated. Perhaps commercially, the mycelium is commercially produced, so you could get a large volume of it in the wake of a fire to try and apply it to the land and remediate these toxins. So these are just a few of the species that might be good for the job. Oyster mushroom is kind of the flagship bioremediation mushroom. Pleurotus austratus, I'd definitely say it's more of a species complex than an individual species. Here you can see got a standard elm or tree oyster up here. We have the pink oyster, which goes by a host of names, including the Dejamor, Cajor sajor, Selmeo semestris. It's a tropical that's native to Florida, anywhere to Malaysia, and superheat tolerant mushroom, so definitely a good choice for summer months. We also have the Citronopelius, the golden oyster, which is a Russo-Siberian kind of Arctic potential one there, kind of a medium temperature. But basically, there's a bunch of different oysters that can fit your climate. Perhaps best suited to breakdown of persistent organic pollutants, but also functional in several types of heavy metal uptake. And while it's not seen in literature, I was able to get a significant barium reduction using it. The phoenix oyster, the pulmonarius, it's less documented in terms of heavy metal uptake, but I think it's important because it can grow on coniferous wood, that's like softwoods, like pines, whereas the other oysters typically can't rise to that job. So if you're in a situation where you're in a conifer woods and that's the material you have to work with, the phoenix oyster is going to be a go-to. From experience, after a fire event, there's a lot of wood chips going around because people are clearing out a lot of dead wood. And so it's a great resource as a material to work from. So turkey tail mushroom, not so functional in uptake of heavy metals, but it's definitely functional in degradation of persistent organic pollutants. It's a great one because it's a super aggressive mycelium. It's can put up with a lot of contamination. The mycelium is aggressive, super hard. So it's a good go-to for especially beginning cultivators. Tiger sawgill, not a commonly known mushroom, but it has some unique traits. Specifically, it's the only mushroom that I found in my reading that is functional in chromium uptake, and chromium is definitely a contaminant that's present in the environment after a fire event around vehicles. It's another super aggressive mycelium, very, very, very tough. It also can withstand soggy conditions. It's also unique because it can grow on willow wood. From my reading, haven't tried it, but it's a way that we could have a twofold bioremediation effect in aquatic situations. Willow is already used as a bioremediation plant, and if you were able to perhaps use it as a uptake tool in our waterways, and then to secondfold chip that willow wood and grow the tiger sawgill on there, you might be able to get a further reduction, a further concentration of the contaminants, the heavy metals in that waterway. Just a concept. Meadow mushroom, so this is Agaricus campestris. This is the wild version of the button mushroom that's commonly cultivated. Functional in a couple of heavy metal ways, great for pastured areas. In that same vein, shaggy mane, functional in some heavy metal uptake, and also well suited to pasture type areas. So can grow it on sawdust and can inoculate it into grassy areas. So what I do as my Butte remediation program, first thing is go out and test soils and find out what's where, what kind of contaminants we're seeing in the environment. A lot of this up until now hasn't really been available. And while more research is being done, at the time that I launched this project, not a lot was understood about fire-borne contamination. Second step was just provide people with help. Sometimes it was literally just scraping burnt material off the ground so I could get in and install fungal infused materials. Other times it was about putting in stopgaps to protect waterways. What I've done through this project is kind of innovate ways that I think we could rapidly deploy materials, augment some of the erosion control materials that we're already putting into place, and do just a little bit better by immobilizing heavy metals and breaking down persistent organic pollutants. And then just trying to see before and after what's happening. Are we getting success? Is fungal bioremediation a functional thing? So again, just go out, test the soils. This is at considerable expense. So when we're testing for polycyclic aromatic hydrocarbons, benzene, dioxins, and heavy metals, it's about $1,000 per sample. So I was limited in the scope that I could do there with the funding I had. Had good success getting fungi to grow on contaminated soils. Also harvested the mushrooms themselves when they cropped up on the site, dried them, sent them to the lab for heavy metal composition to see if we were getting that translocation, that streaming of heavy metals up into the above portion of the mushroom itself, and then resampled the sites to see if we were getting to where we wanted with those heavy metals and persistent organic pollutants. So what does it look like? So in the wake of the campfire, for context, they deployed 300,000 linear feet of these wattles, these straw tubes. And so it was this Herculean task. I mean, each one of these straw tubes would go to Home Depot or something. 25-foot section, I believe, costs about $30. You can get them in bulk, but it doesn't bring the price down too terribly much. And so we're already doing this huge outpouring of effort and materials that are ultimately, in the end, scraped up, taken to the dump with everything else, because that's our solution to wildfires. And if we were to fungally infuse these, perhaps we could be reducing the toxicity of the ash coming out of this situation. And again, biologically immobilizing heavy metals. So some of the work I do is inoculating these wattles. So here you can see pink oyster growing out of one of what they call the metal socks, which commonly use wood chips on the interior. Moreover, I've been working with this burlap jute material. So you can make your own wattles out of them. You can make burlap sacks out of them. You can have a mycelial mat that you're ready to roll out. So what I've been doing is I take this burlap jute material and I soak it overnight in a solution of water and calcium hydroxide. Calcium hydroxide, there's really nice chemical grade stuff that's a little bit more expensive, or you can use the cheapo Home Depot type S lime that you find in the masonry section. Basically what this does is it raises the pH up in the material so high that it kills anything that might be living in there. And then when you drain off the water, that calcium becomes a source of nutrition for the fungus. And then I take that sterilized burlap material, I roll it out on a table, and I use typically leftover material from my own mushroom farm, and I roll it back up in this burlap tube and sits there for 2 weeks. The mushroom grows through that material, and then you have this ready to roll out or twist up or make burlap sacks mycelial mat, this fabric that is infused with fungi. And then here in this last corner, you can see what I'm doing here is I'm taking an already grown out mycelial mat. So this has mushroom growing all through it. I've got a newly sterilized burlap, and I'm rolling it up, kind of doubling down my material so I can make more and make more and make more. So with very little infrastructure and no heat, no fire involved, no sterilization other than chemical sterilization, you can grow a very large amount of this mycelial fabric. And so that's kind of what I've settled on as kind of the best possible solution for readily deployable mycelium in a fire event. So as I started out on this program, I just put it out to folks that I know were getting left behind, and this is what I found. This is nearly a year after the campfire event, and all this is still here out in the environment, and we were coming up on another rain event. So here, this is a whole RV, had batteries in it, had solar panels on it, and everything a year later just sitting there in a pile of ash. Burned vehicles, several burned vehicles on this particular site. So the first thing I targeted was that RV. It was right uphill from chicken pen and some goats. And so with an impending yet another winter coming, I wanted to try and isolate this material and try and mitigate whatever environmental pollutants we found in it. So I went through this, sampled 5 different sites in the ash footprint. And this is what we come up with for heavy metals. So what you're looking at here is on the far left side. This is all the different heavy metals that they typically test for in that CAM17, that 17 heavy metals that Cal OES is looking at. And the results here, this is milligrams per kilogram. So very, very small quantities. Milligrams per kilogram is also parts per million. So 2 ways of talking about the same number. So I used the Canadian standard for safe and allowable. EPA has these really goofy standards where depending on whether you live in the city or if it's an agricultural setting, there can be a 10-fold difference in what's considered safe and allowable, whereas the Canadians have set forward a very much more hard-line standard. So that's what I use to measure success and what's safe and allowable. So emboldened in red is the safe and allowable standard set forth by the Canadian board. And so here we can see barium slightly above allowable level. I mean, chromium, we're talking nearly 6 times safe and allowable. You know, copper's through the roof, which is not a huge concern for health and human safety, but it is a germination inhibitor for a lot of plants. And so it keeps regrowth, healthy regrowth from happening. And consistently, we see way too much zinc in the environment, and that's going to be like galvanized stuff. So in the best of all worlds, these are the fungi that we would use to uptake these heavy metals. Because I was rebuilding my farm and trying to keep my farm in production at the same time, I wasn't able to grow a lot of these fungi for this purpose. And it's noteworthy, Russula, it's a mycorrhizal fungus, can't grow it anyhow. But if we were trying to custom grow a fungi for this job, that's what we would use. So brought in these fungal materials, mushroom mycelium, that jute material, and mulched everything in straw. Again, just trying to encapsulate, keep the movement of heavy metals and toxins, and potentially reduce the toxicity of the ash over time. And we had some success. Mushrooms definitely did sprout from the site, but it's a hard living to make for a fungus growing on toxic ash. So here we are looking at before and after from that site. So they're not all successes, certainly. Barium, nearly threefold reduction. Chromium, a threefold reduction. Copper, significant reduction there. Lead, there's a heavy drop in lead, from 48 parts per million to 19, call that a win. And again, the zinc was cut down significantly. They're not all wins though. It's worth noting over time, the antimony went up. These sampling errors could be that in between the fungal enzymes and the acidity generated by rain and degradation, that it liberates more nutrient or more heavy metals over time. It's also conceivable when I resampled, I happened to catch a flake of soil that had a large amount of antimony in it. And so that's a lot of the unanswered questions and problems, I think, with what I'm doing and not having replicates and ways of knowing whether just natural attenuation, natural degradation would do the job. What I do count as a win in this particular site is the reduction of dioxins. So dioxins are incredibly expensive to test for, so we only test for one, this TCDD. Because it is so expensive, there's this understanding that if you're sampling for one dioxin, there's probably other types of dioxin that are present. And so they report them in these toxic equivalency factors, or different TEFs or TECs, they call them. So while this is reported in picograms per gram, again, very small quantities, they report it in these TEFs. And so the beginning value before fungi were applied is 8. So we got 8 TEFs for this site. After the fungal treatment, we have a near 8-fold reduction. We're down to 1.6 TEFs, TECs, whatever you want to call it. So for a molecule that typically left untouched will last 8 years in the environment and definitely presents a risk for hyperaccumulation and risk of cancer, this is we can call that a success. So these are mushrooms harvested from the site, and we're looking at the heavy metal content. Again, milligrams per kilogram, so it's parts per million, very, very small quantities, and it's in a dried mushroom. None of these values are above normal. The barium, perhaps, if you were to eat a ton of this mushroom in one day, could present a risk, but all these values are way below what you would expect, honestly. And so we are not getting a lot of movement of heavy metals into the mushroom itself. It's just not happening. So second site here is burned vehicles. So I sampled several sites from basically underneath these vehicles, applied our similar fungal landscaping, mycelium, jute landscape fabric, wattles. So this is what we're looking at in terms of the metals that are above safe and allowable levels. Again, zinc through the roof. Lead's not great, but not, deplorable. Again, barium above that level. Again, the mushrooms in the best of all possible worlds, if we could grow these out and apply them to the landscape. Nothing to say these fungi are going to persist in the environment, especially under hot conditions that our summers tend to be. The blue mushroom here for nickel and shaggy parasol, they're not going to tolerate high temperatures, so that's a restriction for mycoremediation. Okay, before and after. So in this one, conversely, we have a significant reduction in antimony, huge drop in arsenic, though that wasn't above allowable levels. The barium, great, 3-fold reduction, we love that. Only a minor reduction in zinc. It doesn't get us to where we want to be, but it is something. And a 3-fold reduction in lead, which is definitely noteworthy. So this is another site. There was a burned RV out in an orchard. This is a situation where helped the landowner just scrape up the last of the debris. We got the frame out of there, got all the material scraped up, and started to put a stopgap in. This is right above a tributary to one of our local creeks, and so definitely worth paying attention to. So yeah, again, antimony above that safe and allowable level. Lead is significant, 3 times the safe and allowable level. And again, looking at zinc. Installed this fungal landscaping and got pretty good success at getting fungi to grow out of the contaminated soil. It's always tough. You're trying to draw this line of supporting the fungus and getting it to be productive, but you don't want to put down so much nutritious material that it doesn't want to grow into the toxic ash. And that's, I think, at the crux of the mycoremediation conundrum here. So again, before and after, significant reduction of antimony. Barium stepped up a little bit, again, disconcerting, but real. Lead, no change. And a step up in zinc. So not exciting results, honestly. Again, it can be sampling error, it can be liberation of more heavy metals over time. It's just hard to say. So another thing in this site is that we saw polycyclic aromatic hydrocarbons, those PAHs we talked about earlier. Each one of these on their own are not really at a level that really is terrifying, but if you take them cumulatively, they do present a risk. And as a result, they were totally degraded. So from before and after, we went from these values here to absolutely none detected. Could it be natural degradation? Possibly, but again, these are pretty recalcitrant, pretty tough molecules. Again, the mushroom from the site, barium content is a little bit higher on this one. That one might be significant. So we're getting some uptake of heavy metals, but it's not something that's significant. We're not seeing a bunch of lead move up into the fruit body. We're not seeing a bunch of antimony move up into the fruit body, and the zinc levels are not something that's significant. So again, images of my own farm, some of the work I did at my own place most recently. Chromium, again, copper's through the roof, not a huge concern for health and human safety, but would definitely prevent germination of seed and regeneration. The lead is significant, and again, zinc. So what we did for the site after the vehicle's removed, all that debris is scraped up for the most part. I mean, we still got flakes of aluminum hanging out in there. Grow the fungus out on the land, getting relatively good soil contact, aggregation of the dirt there on the mushroom itself, getting good mycelial growth through black dirt. So in this one, I did sample for aluminum, a slight reduction. Don't call it a win. Threefold reduction to antimony, that works for me. A great reduction in copper, that gets you back into the safe allowable level, but step up in the chromium. Twofold reduction in zinc. It's not a complete win, but it's a successful effort. Here's the mushroom from that site. So we are getting translocation of aluminum up into the fruit body. I don't have a baseline to bounce this off of in terms of what safe and allowable levels are for food, which is what I'm kind of comparing it to. I mean, this is the mushroom itself, again, the above ground portion, but we are getting a movement of aluminum up into the fruit body itself. So this is how things are now. We've seen an absolutely horrific wildfire year here in California and across the country. We need to find ways that we can mitigate the pollution generated by these fires, and fungi just may be one tool in the toolbox to make things better. I've made a point of saying that it's not a silver bullet. Don't believe all the hype. But at the same time, there are some successes involved. And if we can biologically immobilize the heavy metals that are coming off these fire events, as well as potentially reduce the toxicity of the ash itself by breaking down these persistent organic pollutants, that might be a way that we can help mitigate the impact on the environment moving forward. There's more videos to come. I'm going to do a low-tech mushroom cultivation video. I'm going to do another kind of overview of how we install sites at a farm that was recently destroyed in the Northwest Complex fire. And so please stay tuned to find more content from this feed and help your neighbors. Thank you very much.